Today's video, we will review the major fates of pyruvate, enzymes involved, products formed, and terminology that will show up on the exam. So pyruvate in an abundance of oxygen will make acetyl-CoA, which enters into the citric acid cycle to produce energy. But now, pyruvate converts to acetyl-CoA through the pyruvate dehydrogenase complex enzyme, and the products are NADH, so this is an energy capture step as well as an oxidation step where the metabolite is being oxidized, but NAD plus is being reduced, okay? That's an exam question. And we produce carbon dioxide, which means this is also a decarboxylation step because the metabolite, the intermediate, is losing this CO2 group, okay? It is not carboxylation step. Okay, so as a result of these two products, we also call this step the oxidative decarboxylation step. And this is also a step that might be referred to as one containing transacylation. So this is decarboxylation. An example of carboxylation step would be pyruvate to oxaloacetate. And this is catalyzed by pyruvate carboxylase. That means the intermediate is gaining a CO2 group, so we must have inputted carbon dioxide, and in this we also inputted an ATP. So this is pyruvate carboxylase. Okay, not decarboxylation here because we are inputting CO2. And this is also called an anaplerotic reaction, where we replenish the intermediates of a citric acid cycle. Okay, this is what happens when we have an abundance of oxygen, because as the citric acid cycle goes on, the NADH produced will have to become reoxidized back to NAD, so it can go from glycolysis back to pyruvate. However, recall that in oxidative phosphorylation, there is a terminal oxygen acceptor. What if we are doing strenuous exercise, or what if we're growing yeast under low oxygen conditions? Then we wouldn't have that many oxygens to do oxidative phosphorylation. So that means NADH would have trouble reoxidizing back to NAD, so glycolysis would stop in that case. That's not good. So in low oxygen conditions, in strenuous exercise, as the exam calls it, we use anaerobic fates of pyruvate. So in animals, that would be converting pyruvate to lactate and thereby also converting NADH, oxidizing it into NAD. Or we can do pyruvate to ethanol, and that occurs when growing yeast in low oxygen concentrations, which will also oxidize NADH back to NAD. So this is a substitute for oxidative phosphorylation in low oxygen conditions. We do get a decarboxylation step in pyruvate to ethanol, and lactate can actually convert back to pyruvate, and that would just be the reversible reaction, so that would spit out NADH, once again an energy capture step. All right, another important note for the exam. Here's a question for you. What are some sources for acetyl-CoA to enter into the citric acid cycle? So what are ways that the acetyl-CoA can be produced so that it can enter into the citric acid cycle? So one way would be oxidative decarboxylation, okay? So carboxylation of pyruvate would not be the right answer because it doesn't make acetyl-CoA, but decarboxylation of pyruvate does make acetyl-CoA. You can also make acetyl-CoA via the catabolism of fatty acids and amino acids. So those are reactions that make acetyl-CoA, whereas for glucose, acetyl-CoA is made from pyruvate via glycolysis. Okay. So just so you know, fatty acids and amino acids do not do glycolysis. They have other pathways to make acetyl-CoA. whereas for glucose, we use glycolysis to make acetyl-CoA.